Hi guys, Shweta Dash is welcoming you at the Pharmacy Crash Course division in the Knowledge Club online. At previous episode, we learned about the cardiac muscle structure. Today, we will learn about the cardiac muscle action potential process. Before going to the action potential process, we have to learn about the materials, which materials are directly involved into the action potential process. These materials are very important part of the action potential process. I am going through some very short videos for the action potential section. Don't skip the lectures. If you skip the lectures, you can miss a valuable points in between lectures for your exam. So stay tuned after the end. Today's lesson on the ion channels. Ion channels are proteins that change shape in response to different stimuli to either allow or prevent the movement of specific ions across a membrane. They are said to be selectively permeable. Stimuli which can either come from outside the cell or from within the cell can include the binding of a specific molecule to a receptor on the channel, also known as ligand-gated ion channels. Ligand binding causes a conformational change in the structure of the channel protein that ultimately leads to the opening of the channel gate and subsequent ion flux across the plasma, or a change in membrane potential around the channel detected by a sensor, also known as voltage-gated ion channels, and can act to open or close to the channel. The pore formed by an ion channel is aqueous or water-filled and allows the ion to rapidly travel across the membrane. Ion channels can be selective for specific ions, so there are Na+ means sodium ion, potassium ion, calcium ion or chlorine ion specific channels. They can also be specific for a certain charge of ions, that is positive or negative. Some of the most important ion channels involved in the cardiac action potentials are describing here. Number 1. Hyperpolarization activated cyclic nucleotide gated or HCN channels. Hyperpolarization activated cyclic nucleotide gated HCN channels are integral membrane proteins that serve as non-selective voltage gated ion cation channels in the plasma membranes of heart and brain cells. HCN channels are sometimes referred to as pacemaker channels because they help to generate rhythmic activity within groups of heart and brain cells. HCN channels are encoded by 4 genes HCN1, 2, 3, 4 and are widely expressed throughout the heart and located mainly in pacemaker cells. These channels become active at very negative membrane potentials and allow for the passage of both sodium ion and potassium ion into the cell. This movement is known as a funny current. It's denoted by the IF. These poorly selective cation positively charged ions channels conduct more current as the membrane potential becomes more negative, means hyperpolarized. The activity of these channels in the SAN means SA node cells causes the membrane potential to depolarize slowly, and so they are thought to be responsible for the pacemaker potential. Sympathetic nerves directly affect these channels, resulting in an increased heart rate. HCN4 is the main isoform expressed in the sinoatrial node, but low levels of HCN1 and HCN2 have also been reported. The current through HCN channels, called the pacemaker current IF, plays a key role in the generation and modulation of cardiac rhythmicity, as they are responsible for the spontaneous depolarization in pacemaker action potentials in the heart. HCN4 isoforms are regulated by CCAMP and CAMP, and these molecules are agonists at IF. Second type of ion channel is the sodium channel. These sodium channels are voltage dependent, opening rapidly due to depolarization of the membrane, which usually occurs from neighboring cells through gap junctions. They allow for a rapid flow of sodium into the cell, depolarizing the membrane completely and initiating an action potential. As the membrane potential increases, these channels then close and lock, become inactive. Due to the rapid influx sodium ions, steep phase 0 in action potential waveform activation and inactivation of these channels happens almost at exactly the same time. During the inactivation state, Na+ cannot pass through absolutely refractory period. However, they begin to recover from inactivation as the membrane potential becomes more negative relative refractory period. Third type of ion channel is the potassium channel. Cardiac potassium channels are membrane spanning proteins that allow the passive movement of potassium ions across the cell membrane along its electrochemical gradient. They regulate the resting membrane potential, the frequency of pacemaker cells, and the shape and duration of the cardiac action potential. Additionally, they have been recognized as potential targets for the actions of neurotransmitters and hormones and class 3 antiarrhythmic drugs that prolong the action potential duration APD and refractoriness and have to found effective to prevent or suppress cardiac arrhythmias. The two main types of potassium channels in cardiac cells are inward rectifiers and voltage-gated potassium channels. Inwardly rectifying potassium channels, KIR, favor the flow of potassium into the cell. This influx of potassium, however, is larger when the membrane potential is more negative than the equilibrium potential for K+, means 19 MV voltage. As the membrane potential becomes more positive, that is during cell stimulation from a neighboring cell, the flow of potassium into the cell via the KIR decreases. Therefore, KIR is responsible for maintaining the resting membrane potential and initiating the depolarization phase. However, as the membrane potential continues to become more positive, the channel begins to allow the passage of potassium out of the cell. This outward flow of potassium ions at the more positive membrane potential means that the KIR can also aid the final stages of repolarization. The voltage-gated potassium channels KV are activated by depolarization. The currents produced by these channels include the transient out potassium current I21. This current has two components. Both components activate rapidly, but I2 first inactivates more rapidly than I2 slow. These currents contribute to the early repolarization phase, means phase 1 of the action potential. Another form of voltage-gated potassium channels are the delayed rectifier potassium channels. These channels carry potassium currents which are responsible for the plateau phase of the action potentials and are named based on the speed at which they activate. Slowly activating IKS, rapidly activating IKR, and ultra rapidly activating IKUR. The last type of ion channels is the calcium channel. There are two voltage-gated calcium channels within cardiac muscle. L-type calcium channels, L denoted for the long-lasting, and T-type calcium channels, T for transient, means short. L-type channels are more common and are most densely populated within the T-tubule membrane of ventricular cells, whereas the T-type channels are found mainly within atrial and pacemaker cells, but still to a lesser degree than L-type channels. T-type calcium channels function to control the pacemaking activity of the SA node within the heart and relay rapid action potential within the thalamus. These channels allow for continuous rhythmic bursts that control the SA node of the heart. This is important in the aforementioned depolarization events in the pacemaking activity of the sinoatrial or SA node in the heart and in the neuron relays of the thalamus so that quick transmission of action potentials can occur. This is very important for the heart when stimulated by the sympathetic nervous system that causes the heart rate to increase. In that, not only does the T-type calcium channel provide an extra depolarization punch in addition to the voltage-gated sodium channels to cause a stronger depolarization, but it also helps provide a quicker depolarization of the cardiac cells. The L-type calcium channel, also known as dihydropyridine channel or DHP channel, is part of the high voltage activated family of voltage dependent calcium channel. In cardiac myocytes, the L-type calcium channel passes inward calcium ion currents and triggers calcium release from the sarcoplasmic reticulum by activating ryanodine receptor 2 RY-R2 calcium-induced calcium release. Phosphorylation of these channels increases their permeability to calcium and increases the contractility of their respective cardiac myocytes. L-type channels are activated by more positive membrane potentials take longer to open and remain open longer than T-type channels. This means that the T-type channels contribute more to depolarization in phase 0, whereas L-type channels contribute to the plateau, the phase 2. Before ending today's episode, if you like this video, please like, share and comment freely. Don't forget to subscribe our channel. If you already subscribed, then thanks a lot. And after subscribing our channel, please click on the bell button. After clicking the bell button, you will get our updates regularly. One announcement here. If you or your family members have interest in Indian history, then you can follow our another session, the Itihash Katha, where you can know the true stories of the Indian history, where the narrator name is Mr. Soumitra Banerjee. I am coming with another episode very shortly. Till then, goodbye.